So in principle of animal model is how to make the animal like a human condition. So and as we know that rodent is most popular animal experiment. So mouse is like an icon of animal model. But actually, there is the wide variety of animal models. As we know that like um, elegant, this is roundworm actually, and then like Drosophila and rodents and also rabbit. Yeah, it's very common animal for the animal experiments or animal model. But the mice, rats, and rabbit is the most popular animal model. Why? As you know, that rodent is the most popular animal experiment because the rodent is more effective, efficient, and also representative as the human model. If in some report from European Union that mice and rat is the most popular animal for animal experiments, rather than monkey, dog, also snake, and also goat and another animal. And as we know that in rodent is very effective to represent the human condition in short periods. If we compare between rat age with the human age, for gestational time, they need only 21 days. As we know, in human, we need 9 months. So in 21 days, we can observe gestational period like in human process. And also in rats, only 27 days they reach until toddler age. And then only 300 days they reach until adult condition, and then we only need until 600 days we get the progressive aging condition. So even we make the toxicity test for short period, medium period, or long period, we can assume or we can make the model for whole life condition in human. In acute toxicity test, subchronic even in 28 days or 90 days, and teratogenicity in gestational period, and also in pharmacokinetic or biodistribution. In short period, we can assume in long period in human. That's why the rodent is very popular for animal experiment. And as we know that several methods for animal model, actually not only for, and there is so many methods for animal model, but the most common for animal model is one is genetic modified or gene modified, and the second one is chemical induced, using some chemical, and the third one is by surgery, and the fourth one by diet, and many kind of any method like environment condition, stress condition, and also many kind of method. Actually, there is so many kind of method to make animal model. We want to remind us about the genetic modified in rodent. Actually, this is the most ideal to make animal model, but actually very complicated if we make by ourselves. As we know that in modified rodent, there is some knockout, knock in, and also conditioned genetic. As we have how to modify the gene interest. Is in wild type mouse and in constitutive knock mouse, the gene interest will be delayed. We call it deletion of gene. And also not only in general appearance, but sometimes the researcher, they would like to delete the gene interest specifically in the tissue, for example, internal organ or external organ. But sometimes not only by modified genetically, but some inducer like chemical, biological compound, or stress, and many kind of inducer can shift the normal gene to be a deletion gene. So the appearance also will be changed after the gene will be changed. So, and then the the result of the knockout, knock-in, or conditional genetic modified, we call transgenic mouse. It's very expensive, and in Indonesia, we a little bit difficult to get this one. There's the difference between knockout and knock-in comparison. That the definition is knock-in. It's mean we insert some new gene. So the result, we got the transgenic organism with new character. But in knockout, we delete or deletion the gene. So some appearance, some character will be lost. So it just depends on our research, depend on our hypothesis, or depend on our topic, which one we need to make the genetic modified in our animal. I will give the one of example, the genetic modified. This is the C50 plague 6 mouse with knockout the special gene and IAEL1 DNA glycosylase. So the knockout mouse will be developed to be obesity mouse with high number of white adipose tissue, especially in the inguinal part and also internal organ. So no need to special treatment, no need to special induce, or no need to special diet to make obesity. Even they give with normal diet, the knockout mice will be developed or deposit the fat more efficient than normal mouse. This is the benefit of knockout or transgenic mouse. And also some of animal, not only insert by animal gene or deletion the animal gene also, but some of them they insert by human gene, like now coming popular, the mouse insert by human ACE2 or angiotensin converting enzyme type 2, especially for SARS-CoV-2 experiment. So this animal is very, very compatible or very, very ideal to show the character if the mouse infected by SARS-CoV-2. And this is one of report if some animal or mouse, rat, insert by human DNA, the animal gets bigger brain in mouse. So some neurological experiment will be developed in mouse experiment more ideal. But no need to worry. We just order from the company to get the transgenic mouse, especially there is some company internationally, Charles River, Jackson Laboratory, Biolasco, IDNA. There is many company provide the transgenic mouse. So we just order the type, the animal, which one we need in our experiment. And the most popular method is chemical induced. Chemical induced is very, very common because most of researchers can do by themselves, like streptozotocin. As we know that streptozotocin is chemical to induce for diabetic, even diabetic type 1, diabetic type 2, we can induce by different strategy, and also dimethylbenzoanthracene to induce like breast cancer, and also squamous cell carcinoma, and some kind of digestive system cancer. We also apply dimethylbenzoanthracene with different strategy also. And also monosodium iodoacetate, usually we use for osteoarthritis induced in rodent especially, and better result in guinea pig, and also rotenone to induce the Alzheimer or neurological disorder. Degalactose, usually we use to induce the aging, and phenyl cyclohexidine deepoxide. Actually, so many kind of chemical induced. Yeah, we only searching in journal, and we follow the instruction, follow the dose, follow the administration, and we would like to use what for this method chemical induced for our results. So the strategic is we just searching on internet, on the journal or on the report, scientific report, the chemical induced how to administer, how to make the dose, how to injection, and the strategic of chemical induced, we only check on internet from the journal, so we can follow the instruction and we can running our result. And the surgery method is cheaper, actually cheaper than chemical induced, than modified genetic, but need more skill and experience to do that, because in surgery we have to make the new condition with the physical interference, like a modeling result is more uniform than induced model, because we can make sure that successfully of surgery by physical, we can make sure that the result will be more uniform. But some side effect usually show after surgery. For example, if we make the bile duct ligation in our purpose to make the liver injury, but side effect, the animal will be diarrhea with the lipid proportion higher than normal, for example, like that. If we make some neurological sign, the side effect is the animal is difficult to reach the food, to reach the water. So we have to make a special condition for animal during the surgery method. And also we need the tools, we need the facility, is more sterile condition, because we will open some part of the body and then we expose to the environment, and maybe the environment contains so many contamination. So we need some special facility to perform the surgery method. So I will share several activity in our research center. We make the osteoporosis condition in animal. Actually, will be a little bit long if we wait the animal getting AIDS and getting make osteoporosis. So by surgery method, we can induce some animal with the osteoporosis condition. The method is we remove the ovary. As we know that ovary will be release some estrogen, high number, high concentration estrogen, and estrogen is stimulate the osteoblast. So in theoretically, if we omit, if we take the ovary, the estrogen will be not produced like a normal. They will lose high number of estrogen concentration, like in menopause condition. So in by time, the density of bone and density of skeletal in the whole body, they will be reduced, because they have to absorb the calcium to make the new metabolism. So the density of bone will be decreased gradually. So maybe after 1 month or 2 months after ovarectomy, we can get the osteoporosis condition. To make sure our method is success, usually we confirm by what's called uterus size getting more than normal, because hormonal condition not running well. So the uterus getting smaller, sometimes atresia. This our standard to know that our modeling is success. After we success to get the osteoporosis model, we give the treatment, and then we check the condition of bone matrix by histological, by X-ray, and several methods. We can analyze the bone matrix. For example, in normal condition, we can get the more dense of bone matrix, but in ovarectomy, the number of bone matrix can reduce significantly. So our model of osteoporosis is success to do for experiments. For another example, we perform the brain ischemia, for example, for stroke model. Yeah, as we know that brain ischemia or stroke model is lack of oxygen, lack of blood come inside to the brain in many pathway. For example, in blood stagnation, obstruction, or blood or bleeding inside in the cerebri. But in models, we can do by the ligation in source of the blood vessel who release the blood, who send the blood to the brain. In here is called common carotid artery. We can make the left common carotid artery ligation only, we can make the right side, or we can make the both. So we can get the different severity of the brain ischemia. And the result we can see like this. If we make the only one side of ligation, we only get the one side of lesion in the brain. We call the necrotic area in here. Then we can treat it, this animal, they can recover or not. This especially for neurological nutrition, neurological vitamin, or any kind of neurological supporting therapy. And sometimes we combine between surgery and chemical induced. For example, right now is coming popular also, and the acute respiratory distress syndrome. This is our experiment. We try to open the tracheal part. After we get the tracheal, we inject the tracheal by lipopolysaccharide. This lipopolysaccharide is come from E. coli bacterial compound, and then this chemical will be make the lung injury. We call acute respiratory distress syndrome. This condition is similar with the COVID-19 condition, so that's why this is becoming popular right now. Sorry, and this is the result. By cross pathology, we can see after day 5 and day 14, after injection the LPS or instillation tracheal, in positive control, for example, we found so many bloody area, we call pneumonia, but in some treatment, we can see different result. In T1, they very small effect, and T10 maybe moderate, and T100 maybe more severe than other treatment. And also in day 14, actually a similar condition with day 5, but the situation is more severe than day 5. And also by this experiment, we can check the percent edema in this formula, also already known, formulated by Parker and Townsley. So we can compare between control and treatment. And also lung capacity. Lung capacity usually we measure by histological analysis like this. This lung capacity, it's mean how many lung can receive the air for the breath. We can imagine that in positive control, the number of alveoli, we call alveoli in the room inside in the lung, very few. So maybe 50% then air cannot come inside to the lung. And this is the negative control in normal condition, especially like this. But in several different treatment, we can see T1 is better than T2, and also T2 is better than T3. So we can assume that the T1 is effective to recover the acute respiratory distress syndrome. So this is the way to analyze the animal model for experiments. But actually, so many surgery procedure for animal model like hypertension. Usually we make hypertension with the renal artery ligation, because as we know that if the ren or kidney, they doesn't get blood, they will release some renin enzyme, and renin enzyme will be influence the heart to work more. So hypertension will be occur. And also neurectomy. For example, we cut some neuron in leg or in some part of animal, and then we can make the new treatment to neurological nutrition, for example. And then we can make the gangrene wound, athymic rodent by surgery. We remove the thymus in young animal. And also we can make the push-to digestive system, bile duct stagnation, like the calculi in the bile duct. And then we can make the myocardial infection by ligation in the coronary artery. And then hepatectomy, splenectomy, and cancer deposition we can do by surgery. But actually, there is so many sources or so many literature you can find in internet how to perform this surgery. And the important thing is not only how to make the animal models, but the data analysis is very important. And important things are very, very important. For example, in behavior analysis, usually for cognitive response, cognitive quality of our experiment, usually we use YMS and how many time, correct and incorrect. We can calculate qualitatively, and we can use Morris water maze to measure the memory of mice, and also we can use novel object recognition in animal to measure the memory, the quality of neuron of animal. We can do this one. But not only qualitative analysis, but we can do quantitatively. We can use some software. For example, like this open field test. We just record the animal during the observation, and then we apply in the software, so we can get the map, the trajectory. Sorry, the trajectory. We can get the way, so we can analyze how many times he walked in the same way, and also not only the map, but also we can calculate the cumulative distance, absolute bearing, velocity, and also relative turn direction. So the data more objective, because they will calculate by some software. Maybe I just for the information, I run this analysis in the R software. Actually, R software is open source software, so you can download by yourself in internet, and you also can get the instruction how to apply, how to analysis the behavior analysis in our software on internet. Actually, it's not so difficult to perform this analysis. So the data can report more ideal. So more people can understand easily if we make the data more quantitative. So we can make trajectory, similarity, we can analyze the statistical data, and we can analyze any kind of measurement. And gross anatomy histopathology is very, very important to assay the successfully of modeling. But as we know that histological gross anatomy also belong in qualitative data, and not all people can understand easily. For example, if they're not familiar with the histological figure, they will not understand about this, about control, how about in low, how about the height. But by quantitative analysis by software, they can easily to understand, like cell number in millimeter. They can analyze it by quantitative data, and also give the statistical data analysis, and also the cortical bone by spatial staining. They can make the comparison between control and treatment, and color intensity. They can compare between each other, and any kind of analysis by histological. Maybe I will introduce, or maybe some of you already familiar about the image analysis. Actually, so many image analysis for processing in like Photoshop, image metrology, IMG, and cell profiler. But in here, we familiar with IMG, so I will introduce to you briefly about the IMG. IMG is one of software. Actually, is open source software. You can download on internet. You can click IMG and IH curve. So This software is released by NIH in USA, and you can download based on your operating system in your computer, like Mac OS, Linux, Windows, and documentation. After you download, you can get the window like this. Only simple window like this. There is some file, edit, image, and another kind of menu. And then after you get this window, you can analyze your data. For example, you can measure the length. For example, actually difficult to say that this one is bigger than another. But after we measure the length by software, we can get the quantitative data. For example, this follicle, this ovary, after we make the histological section, we can measure how long the length, how long this follicle, how long this one, how long this one. So we can make the statistical data. And also we can compare each other. This is the result, not only length, also we can measure the angle when we make the line. And that's why we can compare between treatment. For example, this one control, treatment 1, treatment 2, treatment 3. So we can compare the statistical data about the size of follicle more precisely and more comparable. And not only length, but sometimes we also have to measure the area, because not compatible if we measure the length like this follicle, because not complete circle in shape. So that's why we have to measure the area. So in this software also we can calculate the area, so we can know the distribution, the area of all follicle, and also we can compare each other between the treatment. And usually not only calculate the length, calculate the area, but also we can check the proportion of glomerulus with the capsule, Bowman capsule, and also proportion of number of necrotic cell, and the capacity of lung, like we discussed before, and also size of red blood cell. Actually, we can calculate more precisely by in this software. And then we can calculate the expression of some protein or some marker. For example, we want to calculate the fibrosis. We just use the percent or area fraction, how many percent this fibrosis expression in one of slide, and also immunohistochemistry. By threshold method, we can calculate more precisely the proportion, the positive cell with the negative area. And also we can make the counter analysis. As we know, in conventional method, we use this tools like conventional. We just push the button and then they record the number, how many cell we check in the slide. But in this software, we can calculate by software, even manually or automatically. I will show you how to counting the automatically in the next slide. But not only the number of cell, we can also the counting the intensity of color. We can measure the redness color in this follicle and another follicle by the software. In mean, it's mean the green value unit, how to calculate the color, the follicle. So we can make the graph, for example, how intense the positive immunohistochemistry between column number 1, number 2, number 3, 4, and 5. So we can calculate the brownish color or brown level color in this slide. So we can make the statistical data more qualitative, more quantitatively, and then we can put the statistical analysis also after we got the quantitative data. And also we can calculate the circularity or roundness in the object, because we can analyze in especially in immunomodulator, immunodeficiency or immunomodulator research. We have to calculate the young cell with the mature cell in spleen. In spleen, for example, mature cell indicate by round-shaped cell with the color with the dark color in cell, but in young cell indicate the maybe a little bit not round shape but some irregular shape. So we can calculate the circularity. As we know that circularity nearly by 1 score is near in round shape, but in near in 0 score is belong in irregular shape. So we can calculate the circularity all of the cell in this feature. And sometimes we have to calculate not only 10 or 100 cells, but maybe 1,000 or more cells. So if we calculate manually like before, it's a little bit difficult. In here, in this software, we can calculate automatically by some menu in this software. You can calculate automatically how many cells in this area, and also how big the cell in this area, size, the color. You can get the score, and also circularity. You can get the score. So in one analysis, you can get at least 3 kind of analysis: size, area, in means, color, it's mean, and then circularity or shape of the object you observe. And not only for microscopy analysis, IMG also common use for calculate or quantification, though, like Western blot data or any kind of STS-based data or dot plot analysis to quantify the band or the dot intensity and also the size. So you can get the data like this, the number of area in pen number 1 like this, pen number 2 like this, number 3 and number 4. So we can make the score, and then we can do statistical analysis. And also even we doesn't have, also we can do the surface plot analysis. This is our research in osteoarthritis in sheep. After we induce some chemical data, we can analyze the surface plot, the roughness or smoothness of the bone in this area. So we just make the area we want to analyze this, and then we check the surface plot, and then we compare the roughness or the smoothness of this area. So the people may be more easy to understand this figure. Then we give this figure. So I think quantitative data is more important in model analysis, and not only for microscopy, but macroscopy we can analyze by in this software. For example, how to measure from the nose until the tail, how big the eyes, how big the ear, and some part in the animal body we can measure only by in this image or in the photo. So no need to make the measurement in animal. And maybe this one is more simple way to do the measurement. And sometimes if we have no ELISA reader, actually it's not ideal, but we have to do the experiment. So absorbance analysis, we can also calculate by the figure. We can just take the photo, the plate, and then we measure the color, and we compare the standard curve. So we can calculate the optical density of the sample. We can calculate the concentration after we transform or we change to the standard curve calculation. So actually this software is very, very useful, not only for histology, but also for macroscopy and also another analysis. This is the way how to calculate the color, so we can compare the color intensity from each sample. Even we doesn't have any ELISA reader. That's all. Maybe not so many can I share to you, but I wish you I get also some information from you, and maybe we can share each other about the animal model, about the analysis, and any kind of experiment. Thank you so much for the time, and maybe I will give back to the MC for the time. Thank you so much, and Assalamualaikum warahmatullahi wabarakatuh.
Waalaikumsalam warahmatullahi wabarakatuh. So thank you to Dr. Subangkit. And yeah, that's all from Dr. Subangkit about animal model in drug discovery research. We got to know things like the wide variety of animal models, several methods for animal model, for instance, like gene-modified, chemical-induced, surgery, and diet. And well, I will open discuss session for those who want to ask directly to Dr. Bangkit. I warmly welcome. So yeah, is there any question? Just raise your hand. Maybe there's no question to Dr. Subangkit, or what? Okay. I think there's no question, Doc. Okay, I want to remind for the students of Biopharmaca summer course that post-tests will be brought forward today. So don't forget to do your post-test. Before we got to the break session, we will watch up about video profile of animal testing laboratory.
Maybe I have question. Yes, Aura. Is it?
Oh, yeah.
Yes, question for Dr. Bangkit. It's very interesting talk. I think Pak Bangkit, Mr. Bangkit, Dr. Bangkit. I'm Wisnu from computer science. We already know. My question is, what actually the potential? I mean, the potential collaboration between computer science and with your field, especially for developing research or develop the software related to the animal model and so on. Thank you.
Yeah. Can I answer directly, Dr. Ms. Aura?
Yes, you can answer that.
Thank you, Dr. Wisnu, for the question. Actually, it's very interesting to collaborate between computer science with the scientific experimental, because like me, I work as veterinarian, also researcher in the animal experiment. We still have many analysis in qualitative data, and that data is depend on the experience of the researcher. So if we can make some software can quantify some data, it will be more helpful for all researchers. For example, like me, I work also as in pathological field. Many data depend on the researcher. I can say good, and maybe another researcher can say not good, because depend on our subjectivity. What's called subjectivity scoring. But if we can make some software to quantify the data, like for histological analysis, and then we just put the picture in that software, and then the software can scoring automatically. So there is no excuse to say good or not, maybe like that. And not only for histological analysis, for example, like I saw you before, many behavior analysis, how to track the animal, how many animal breeds, how many the animal run, how many the animal stay. Right now we just do by scoring qualitative data. We just put the score 1, score 2, score 3 like that. But if some specialist computer like Dr. Wisnu can make the software to quantify the data like behavior analysis and also the histological analysis, macroscopical, and many data by software analysis, maybe the research can more easily to perform, and maybe the researcher can do more confident, because as we know that qualitative data ourselves, sometimes we're not confident with our scoring is good or not, maybe a little confused like that. That's all, Dr. Wisnu. Actually, so many collaboration between computer science and experiment data analysis. And right now in European country, in American country, and also Japan, maybe well, they develop the many software to analyze the experiment data. And that's all, Dr. Wisnu.
Thank you very much, Dr. Bangkit. I think, is there any question in chat room?
Well,
there is one question in chat room. Yes.
Yeah, there's one question from Patricius Atmojo from Atmajaya to Dr. Subangkit.
Yeah.
How to choose animal test or assay for our research?
Okay, thank you, Dr. Patricius, for the question. Yes, actually, this is the basic question, but very important question. How to choose the animal test? How many animal need to use in this experiment? Actually, it's very important things. And thank you for reminding this. Actually, how choose the animal test for assay is depend on our hypothesis. Yeah, first, at least in rodent, for example, they divide into kind of a strain. The one is inbred strain, and the second one is outbred. And as we know that inbred strain, it's mean they will make the homological data if we make some intervention, for example, in modeling. So that's why many modeling experiment, they prefer like use the inbred strain rodent, like C57 black 6 mouse, maybe Levis rat, and many kind of inbred, because when we give the treatment, they will give the uniform data, more uniform data. But different concept, if we want to know the effect of some drug, effect some what's called some biological compound, some chemical induced in human model. For example, we develop some what's called jamu, or many kind of biological compound. For example, we want to know the effect in human population. We cannot use inbred strain because they will give the uniform response. So we usually choose the outbred rodent. In mice, usually we use DDY or PUBC. In rat, we usually use Procdoli or Wistar, because, for example, in 10 animal, we give same treatment, same condition, will have different response and different result. But that's the real condition. What we want to know how it's happened if we give to some population in human. This way to choose the animal. That's the one concept. The second one is we have to know our modeling is reliable or not if we perform in one animal. For example, if we want to make the liver injury by bile duct ligation, as we know, in rat they has no the bile duct what's called bile duct, but in DDY in mice they have bile duct. So that's why even mice is smaller than rat is compatible or is appropriate for bile duct ligation method. And also in some any kind of research in cardiology, usually not only used for rodent, we usually use like dog, for example, because the dog condition in cardiology is quite similar and closely similar with the human. Sometimes we use dog for the cardiological experiment. So, and choosing the animal for appropriate research is need more discussion about how to choose. And the number of animal, actually so many formula to calculate how many number of animal we use for experiments, like Cannon formula like Federal formula, but we have to back to how many data we need to calculate more precisely, especially for statistical analysis. Even in Federal is need for 5 animal per group, but in statistical analysis is not enough. Actually, we can still add more animal to get the appropriate data, to get the ideal data. So actually, it's so wide discussion to choose the animal, kind of animal, to how many select the, how many animal need in one group for experiment. Maybe I wish can answer your question, Dr. Patricius, and success for your experiments. Thank you.
Okay, well, thanks to Dr. Subangkit for answering those kind of questions. And I think the answer quite clear enough. So next, for the committee who've been assigned to the video profile of annual testing laboratory, time is yours.